Hello, welcome to Chapter 15. This lecture will cover your third line of defense, otherwise known as your adaptive immune system. We'll talk about the specificity of this line of your immune system, and we'll also include some discussion on immunizations. First, to begin, we'd like to define what specific immunity and the adaptive line are. The third line of defense is going to be involving your T and B lymphocytes. Specifically, through a dual system and interaction between the B and T lymphocytes, we do find that a stimulation of specific antibody is produced in response to foreign molecules known as antigens. The thing about antibodies is that they are specific to the antigen that stimulated their production. So what this means is that one antigen is produced for every type, one antibody is produced for every type of antigen that your body encounters. So this is going to be one of the features of our specific immune system in the third line, which is specificity. Specificity is going to be about those antibodies that are produced against that specific antigen. They will only interact with that type of antigen that they were produced in response to. In addition to that, during this response mechanism, during this dual system interaction between B and T lymphocytes, we will also find that lymphocytes produce memories. So in this case, you can actually look at this as a typical definition of a memory. They do remember. So we call them memory cells. B memory and T memory lymphocytes basically are going to remember the encounter that they had with that antigen from the very first time. And when they encounter that antigen again, they can actually skip a few steps during the response mechanism that produces antibody, and they can actually respond in a more rapid fashion then. So what this means is that we can make antibody a little quicker the second or third or fourth time your body interacts with this specific antigen. So at this point in time, I actually need to start introducing to you some of the puzzle pieces that go together with this dual system of B and T lymphocytes. So when it comes down to it, we need to first start with cellular receptors. So we're really going to be talking about pieces of protein that protrude from the outside surface of a cell. So we're really going to be looking in a very microscopic fashion at things on the outside of our human body lymphocytes. So first we'll talk about cell receptors or markers. These things are going to allow for specificity and to confirm identity of a cell. Everybody has their own type of cell receptor or markers that basically allow them to be recognized as an individual, not only on a cellular level, but also on a tissue level. So they have major functions, which is going to be to perceive and attach to non-self or foreign molecules. These types of cellular receptors will be found specifically within our immune system cells. You can also find cellular receptors that promote the recognition of self molecules, meaning that they are basically going to be molecules that promote self as a marking. So they basically display that to all body cells. They say, I am self. And then also they can be used for cellular communications, and they can receive and transmit chemical messages between themselves and other cells. They can also be a point of stimulation to aid in cellular development. Specifically, when we look at B cells, we will find that the cell receptors or markers are going to receive signals that actually allow them to proliferate into multiple cells. So let's talk specifically about some of these cellular markers. So first of all, we're going to talk about major histocompatibility complexes, otherwise known as MHC. MHC and HLA, human leukocyte antigen, are interchangeable terms. They're pretty much the same thing, just different ways of looking at these cellular markers that are going to be present on the outside of our cells. So these are going to be found on all cells of the human body except for red blood cells. Red blood cells have their own marking system, which actually make them more universally exchangeable with just blood typings versus MHCs and HLAs. They have a lot to do with tissue typings. Tissue typings are a little bit more complex and a lot less interchangeable than red blood cells or blood. So generally speaking, we have two major types of MHC, or major histocompatibility complex. We have class 1 and we have class 2. Class 1 is going to display unique characteristics of self, so it puts it out there that the cell that is attached to is part of the body. And then it also is going to have a role to play in regulation of immune reactions with cytotoxic T cells. Cytotoxic T cells are going to be a specific type of T lymphocyte that, in addition to other markers, will also display a marker known as CD8. The CD8 marker is going to be a very unique marker only found on cytotoxic T cells. Because of this cytotoxic T cell specificity, we do find that MHC class 1 only regulates those regulates the immune system reactions between cytotoxic T cells. And that's because MHC1 can directly communicate through and with the CD8 marker found on the T cytotoxic cell. So it's a type of specificity of communication between those two types of cell. So one of the ways that I actually encourage you to remember which MHC would actually communicate with which T cell would be by looking at the numbers associated with the classes. So MHC1, the 1, and the 8 in CD8, if you make it into a multiplication table or a multiplication problem, 1 times 8 is 8. When it comes to pairing the MHC class with the type of T cell, 8 is great. And this will make just as much sense when applying it to our next class of MHC, which is the class 2. Class 2 receptors are also going to recognize and react with foreign antigens. They're also going to have a level of promotion of self. But they are located primarily on macrophages and B cells, and they are going to do a lot of interaction between macrophages, B cells, and T helper cells. T helper cells are a type of T lymphocyte, which are known to help the dual system of antibody stimulation. It helps it actually happen. So the involvement of this T helper cell and then brings in the fact that we must discuss its specific marker. Its specific marker is known as the CD4. So sometimes we just call T helper cells CD4s. And because it has a CD4, it specifically communicates that CD4 with the class 2 type of MHC. So again, if we actually make this into a multiplication table, multiplication problem, take out the letters, put in only the numbers, and the MHC2 times the CD4. So in other words, 2 times 4 is 8. Again, 8 is great. That way you know you have the appropriate class matched to the appropriate T cell. Here we have a picture showing us the MHC class 1 and class 2 molecules. They're both made out of polypeptide or protein, and they do protrude from the surface of our cell membranes. So if you can imagine that these would be hanging out of the top or the surface of a body cell or other white blood cells such as macrophages, B cells, other lymphocytes, so forth and so on. You can see that the class 2 MHC is going to be found to have kind of a cup shape at the top. That cup shape at the top, or that hole that's at the top, is actually the location of the presentation that occurs with the MHC2. There's a presentation of process antigen that occurs between MHC2 and T helper. And that location, that opening, is specifically going to be where that occurs. And you can see that the shape of this protein is very much lined up to play a role in that. Some other puzzle pieces that we must put into play here are going to be lymphocyte receptors. Lymphocyte receptors are going to be specific to only lymphocytes, which basically come in the form of B lymphocyte and T lymphocyte. B lymphocytes are going to have B cell receptors, which have the ability to bind free antigens, and T cell receptors are going to bind processed antigens. When it comes down to it, B cell receptors are going to be made out of immunoglobulin. Immunoglobulin is basically the same compound that makes up antibodies. Immunoglobulin is going to be also a B cell receptor. What we'll find out later in this chapter is that B cell receptors are a type of antibody known as IgD, and B cell and T cell receptors are going to be made out of different types of protein. So B cell receptors are made of immunoglobulin. They're going to have a Y-shaped arrangement to them. The top of the Y portion is going to have an antigen binding site, and it is going to be also known as the variable region, because that region will change depending upon the antigen that it binds to. This is what a B cell receptor looks like. A B cell receptor, like I said, the yellow portion here is the variable region. It is going to vary depending upon the antigen that it would bind to, because all antibodies are specific. The variable region is the antigen binding site region. The rest of this region of the antibody that is located in a maroon color here is going to be known as the constant region. The constant region is unchanging, it is constant. Next up, we must talk about the types of antigens. Antigens are basically anything foreign that's not a normal constituent of the body that contains an antigenic determinant or epitope. So there's a piece of it or a part of it that our immune system recognizes as foreign. Antigens have a lot of different types of antigenic determinants. When it comes down to it, we do have 5 major classes of antigens that are special categories. So we have the haptens, which are very small foreign molecules. We have alloantigens, which are going to be cell surface markers of one individual that are antigens to another of the same species. So these are basically going to be antigens of tissue and organ transplantation. Super antigens are those antigens that promote a potent T cell stimulation. It provokes an overwhelming response by the immune system to the point of detriment. Basically, it ends up making our immune system harm us more. Allergens, and super antigen examples are going to be things like Staph aureus, or even in some cases, group A strep pyogenes. Allergens are going to be antigens that provoke an allergic response. And then autoantigens are going to be molecules of your self-tissue which you are not tolerant to any longer. These are going to be the antigens that actually stimulate autoimmunity. So autoimmunity is something we'll cover in another chapter. Antigen processing and presentation is going to be another one of the puzzle pieces that we must discuss before we get into our third line of defense mechanisms. Antigen processing and presentation basically means that an antigen in its current form cannot be seen or recognized or even attached to by a T cell. When it comes to antigen processing, this must be done by specific types of macrophages. known as dendritic cells. So some basic macrophage, but more specifically dendritic cells, are going to be known as APCs. APCs are antigen processing cells that are going to be the first cell to interact with an antigen in the second line of defense. And then it will process it by breaking it down into a more recognizable form by a T cell, and then it will present it on its MHC receptor. Now, in turn, during the activation of our third line of defense, B cells must also be able to antigen process and present. So B cells are going to be another form of APC, or antigen presenting cell. So basically, antigen processing and presentation is going to be how we stimulate the majority of our cell-mediated immunity, which is going to have a direct involvement with T lymphocytes. T lymphocytes act directly against antigen after they have been processed and presented in response with an MHC carrier. So basically, one type of immune cell, like a macrophage or dendritic cell, gobbles it up, processes it, places it out on its MHC, and then goes and shows the T cell. The T cell can then, in that place, respond, not before. There are 3 different types of T cells that we will discuss. The first is going to be the T helper. I did give you a small introduction to the T helper already. T helper cells are the most prevalent type of T cell. They are also known as your CD4 cells because they do have the specific marker of the CD4 on their cell surface. They also are going to help regulate immune system reactions to antigens. And there are 2 varieties of them, T helper 1 and T helper 2. T helper 2 is going to be the one we'll focus on that's going to have everything to do with the activation of our third line of defense. T helper 1 is going to be mainly involved in activation of allergic reactions that are known as hypersensitivity type 1. We will discuss hypersensitivities in the next chapter. When it comes to cytotoxic T cells are known as CD8 cells. CD8 because of the specific marker found on their cell surface. They are known to destroy foreign or abnormal cells by secreting perforins that lyse the cell. So they do have a direct attack associated with them, but only after activation by an antigen-presenting cell. We will discuss this process later in the chapter. Natural killer cells are going to be another variety of T cells. Sometimes they're called NK cells. Sometimes they're called natural killer T cells. Either way, they are a unique type of T cell that lacks specificity. So they lack specificity in the fact that they can actually be associated with the second line of defense. So they are not only going to attack specific cells like your T helper and T cytotoxic are involved with because of that direct and specific activation. Natural killer cells are going to be cells that actually respond generically to viral infection as well as to cancers, as it turns out. T cell maturation must occur before activation. T cell maturation does occur in the thymus, or I'm sorry, with the thymus gland hormones. So basically, T cells are produced by the bone marrow. After they are produced by the bone marrow, they are subjected to a round of hormones secreted by the thymus. After the thymus secretes the hormones upon them, they are then directed to be a particular type of T cell. For example, some become T helper, some become T cytotoxic, so forth and so on. After maturation, they will then undergo activation. Activation is basically the antigen processing and presentation process. We will pick up in Chapter 15, Part 2, with activation of T helpers and the activation of our third line of defense.